Hello students, welcome to Zoology Classroom. In today's class, we are going to know about resting potential, threshold potential, and action potentials. So these topics are very important to understand the functioning of nervous system. So as we have already discussed in previous class, nervous system plays a very important role in coordinating all the other organ systems activities, and nervous system also plays a very important role in understanding what is happening around us and what we have to do. So all these things will be done by this nervous system. So understanding the surroundings, foreseeing the consequences, taking the decisions, and seeing that all those decisions are uh, applied, or all those decisions are what we say, or what we can say, all those decisions are to be followed by other organs in the body. So all these are the functions of the nervous system. And all these activities done by the nervous system will be done in the form of action potential only. So if we understand the concept of action potential, how this action potential arises or generated, and how it is propagated, if we understand these things, it will be easy for us to understand the functioning of nervous system. So the functioning of nervous system is based on this action potential. So potential is nothing but a form of current. We know that the current flows in this electrical wires, like that some form of current in very minute, or what we can say, in very low quantities, some kind of current will be flowing inside these neurons and nervous system. So this is called as action potential. So, and we can measure this action potential with the help of a voltmeter. So in this slide, you can see a voltmeter. This is the voltmeter. So in this voltmeter, a needle will be there, and we can insert two electrodes into the neurons. One electrode enters into the inside of the neuron, that means into the axoplasm or cytoplasm of the neuron, and another electrode will would be placed on the neurolemma or plasma lemma of the neuron. Okay, so one would be on the outside of the membrane, another electrode will be placed inside the membrane into this cytoplasm or axoplasm. So if we place these electrodes, and these two electrodes are connected to voltmeter, so when nothing is happening inside these neurons, minus 70 millivolts of potential or current will be shown in this voltmeter. So when nothing is happening, when the neurons are not doing anything, when the neurons are not in function, when the neurons are not doing their work, minus 70 millivolts of current will be there. This is called as resting potential or resting membrane potential. And when the neuron is in work, when the neuron is doing work, what is the work? Generating the action potential and transferring the action potential to the neighboring neurons. So when the neurons are doing their work, this capacity or this potential will be changed from minus 70 to plus 30 millivolts of potential will be generated. So the change in this potential is called as action potential. So when the neurons are in action, the potential will be changed. When the neuron is not doing work, minus 70 millivolts of potential will be there. So as the neuron is in rest, this minus 70 millivolts of potential is called as resting potential. But when the neuron is in action, when it is working, the minus 70 millivolt of potential will be upgraded to plus 30 millivolts of potential. So the upgradation in this potential is called as action potential. Why? Because this upgraded potential will be generated during the neurons are in action. So that's why it is called as action potential. So this is very important. Okay, so to understand how these potential changes are taking place. So say for example, the neuron at this moment it is in resting position. So potential will be minus 70 millivolts. After some time, it is doing some work. Then the potential will be upscaled to plus 30 millivolts. Later again, it will come to resting potential. So again, from plus 30 millivolts to the potential comes down to minus 70 millivolts. So like this resting potential, again action potential, again it comes to resting potential, and again next time it goes to again action potential. So how these changes are taking place inside this neuron, how these changes are taking place in the potential differences. So this is very, very important. So to understand this thing, so and as I have already told, in this slide we are seeing a voltmeter. In the voltmeter, a needle will be there, and this needle will be showing the readings, and these readings will be mentioned. Say for example, minus 100 to plus 50, like that, plus 50 millivolts. So the speedometer like thing, it is voltmeter is nothing but a speedometer like thing. Speedometer we can see in the vehicles. So in this way, in the same way, the voltmeter also will be showing us the what is the potential present inside this neuron. So this voltmeter shows as the potential present inside the neuron, not on the outside of the neuron. So the value shown in this voltmeter belongs to the potential of the inside of this neuron. Okay. So and another machine also can be used to measure the potential differences inside this neuron, and that machine is called as oscilloscope. So in this oscilloscope, in this thing also, electrodes will be placed, one in inside this neuron, one on the outside of this neuron, and these two electrodes are connected to this oscilloscope machine. And in the oscilloscope machine, we can see the potential differences in the form of a wave-like form, in the wave-like form. So this is just like the ECG machine. So how the ECG machine shows us the functioning of heart. In the same way, this oscilloscope helps us to know the wave formations. And by showing the wave formations, we can understand how this action potential is generating and how it is propagating or transmitting from one place to another place. So two things can be used to measure this potential differences inside the neuron. One is oscilloscope, another one is voltmeter. Okay. So how these differences? So when the neuron is in resting position, so we have been told that when the neuron is in resting position, minus 70 millivolt difference will be there. So how this minus 70 millivolt of potential is generating inside this neuron? So to understand this thing, we have to go back. So in the previous class also, I have told you that inside the neuron, cytoplasm is there, or we can call it as neuroplasm. Okay, neuroplasm or cytoplasm is present inside this neuron. And outside this neuron, extracellular fluid will be there. Okay, so the extracellular fluid is a liquid, and a neuroplasm or cytoplasm is also a liquid. So these two liquids are separated by this neurolemma, or plasma membrane of neuron. Okay, neurolemma or plasma membrane of neuron is separating the extracellular fluid from intracellular fluid. And this intracellular fluid is nothing but the cytoplasm of the neuron. Okay, so in both these liquids, ECF and ICF, ICF is inside, ECF is outside, exterior. So in these two things, many ions will be there, like the sodium ions, potassium ions, chloride ions, and many anions. Anions are negatively charged ions. So A minus means anions, negatively charged ions. So many positively charged ions will be there, and many negatively charged ions will be there, both inside this cell and outside this cell. So these, the combination of these total positive ions and total negative ions, a difference will be there. So mostly all our cytoplasms, all our blood, and all our extracellular fluids, or all type of liquids present inside our body will be in neutral condition. Say for example, in this thing, we are seeing that 150 potassium ions and uh, sodium ions are in 15 millimoles. So 150 millimoles for potassium ions, 15 millimoles sodium ions. So total positive ions quantity is equal to 165 millimoles. So at the same way, total anions and another this my negatively ions are also will be equal to 165 millimoles. So in this way, what I mean to say is that inside the cell, the total positive charged ions quantity is equal to total negatively charged ions. So both will be in neutral condition. The same thing applies to the outside of this cell also. In the extracellular fluid also, many positively charged ions will be there, many negatively charged ions will be there. So the sum of these positively charged and negatively charged ions will be equal to each other. So outside neutral condition is there, inside also neutral condition is there. But one thing is very, very important, that outside both things are equal, inside also both positive negative charged ions are in equal quantity, but there is a difference in between total outside and total inside. Okay, total outside quantity and total inside quantity is not same. In the inside, total positive charged ions are equal to total negatively charged ions. Outside also, total positively charged ions are equal to total negatively charged ions present outside. But the total charge of this inside ions and outside ions will be different. So when the cell is not doing anything, when the neuron is not doing anything, what will be happen? What will be the condition? Is that outside positively charged condition will be there, inside negatively charged condition will be there. Okay, so that means when compared to exterior, interior will be having a more negatively charged condition will be there. So the difference between outside and inside potentials is minus 70 millivolts. So when compared to outside, inside minus 70 millivolts difference will be there. So inside negatively condition will be there, outside positively condition will be there. So these conditions will be changing when the neuron is doing work, or when the neuron is propagating action potential. What will be happen inside minus 70 millivolts condition will be changed to plus 30 millivolts condition. So inside of the cell will go from negatively charged condition to positively charged condition. At the same time, outside positive condition will be turned to negative condition. So when the neuron is doing work, outside becomes negative, inside becomes positive. Again, when the neuron comes to resting position, again outside becomes positive, inside becomes negative. So the positive negative positions will be changing in between these extracellular fluid and intracellular fluid. So the changes in these conditions, the changes in these potential conditions is due to the traveling of sodium and potassium ions across the membrane. So at some time, sodium ions enter inside the cell and potassium ions go out of the cell. And again later, potassiums again they enter into the cell, sodiums go out of the cell. So the sodium ions and potassium ions, these two ions play very, very important role in generating action potentials and again reinstalling the resting potential. So many ions will be there inside and outside the neuron. Inside and outside the neuron, many ions will be there: sodium, potassium, calcium, chloride, anions like bicarbonate, carbonate. So like this, many ions will be there. But out of all those ions, sodium ion and potassium ion are very, very important for the functioning of nervous system or for the functioning of a single neuron. So sodium and potassium ions play a very, very important role in the functioning of neurons or nervous system. So as I have already told, these sodium ions and potassium ions will be moving from inside to outside and from outside to inside. So across the membrane, across the neuromembrane of these neurons, plasma membrane of the neurons, the sodium and potassium ions will be moving here to there to here. So the movement is facilitated by some type of ionic channels. So some type of ionic channels are present on the nerve membrane or plasma membrane of neuron. So these ion channels are of four types. One is ungated ion channels or leaky ion channels. In this ungated leaky ion channel, sodium and potassium ion channels are very, very important ones on the plasma membrane of neuron. Second type of channels are voltage gated channels. These voltage gated channels play very important role during the functioning of neuron, or when the action potential is generated. The action potential is generation is due to the activity of voltage gated channels only. And third type of things are mechanically gated channels. These mechanically gated channels are present on the neurons, which are present on the recepting organs. So say for example, skin is a recepting organ. Skin receives the pressure and temperature and pain. All these informations, all these knowledges, or all these sensations are received by the skin. Like that, in our eyes and in our ears, in all the sensory organs, 5 sensory organs are there. In the sensory organs, the neurons are called as receptors. So the receptors are one type of neurons. All these receptors will be having mechanically gated channels. So these mechanically gated channels, they will be opening due to the mechanical stimuli. So when the mechanical stimuli is applied, these mechanical gated channels will be open, and that will lead to the generation of action potential. And fourth type of channels are ligand gated channels. And these ligand gated channels are present on the synaptic membranes. Presynaptic membrane and postsynaptic membranes will be there. On the postsynaptic membrane, these ligand gated channels will be there. So ligand-gated channels will be present on the postsynaptic membranes. So each neuron will be having presynaptic areas and postsynaptic areas. And on the postsynaptic membranes, these ligand-gated channels will be there. Presynaptic membrane will release neurotransmitters, and postsynaptic membranes will receive those neurotransmitters. Those neurotransmitters will join these ligand-gated channels. So when the neurotransmitter is attached to the postsynaptic membrane, ligand-gated channels will open, and the ionic transfers from inside to outside or from outside to inside will take place in the postsynaptic neuron. So like this, ligand-gated channels are present on postsynaptic membrane. Mechanically gated ion channels are present on the receptors, which are present on our sensory organs. And voltage-gated channels are present on all neurons. All type of neurons will be having voltage-gated channels, and all type of neurons will be having this ungated or leaky channels. So ungated leaky channels, here we can see this always open. Ungated leaky channels will be open all the time. So all the time, some amount of sodium goes from inside to outside, and sometimes it goes from outside to inside. Like the sodium also will be going from out to into out. So this movement will be seen always. So we can see the movement of these ions through these ungated channels always. So they are always open. And voltage gated channels, the second one is voltage gated channels. So these voltage gated channels will be opened only during the stimulus is given to the. When the stimulus is given, during that time only this voltage gated channels will be open. When these are open, then only action potential will be generated. So these two are very, very important in understanding the functioning of nervous system. Ligand gated channels also function in the same way. Mechanical gated channels also will be functioning in the same way, but mechanical gated channels are present on the receptors present in sensory organs. Ligand gated channels are present on the postsynaptic membranes. This is the only difference. So the ligand gated channels functioning, mechanical gated channels functioning also will be similar to voltage gated channels functioning. So the functioning of ligand gated, mechanically gated, voltage gated channels will be the same. But today we are going to know about voltage gated channels. The same thing applies to mechanically gated channels and ligand gated channels. Okay. Next, in this way, so during the action, during the neuron is not doing anything. that means during the resting position inside this cell. This is cytoplasm. This is extracellular fluid. So in the extracellular fluid, more sodium ions will be there. In the intracellular release, in the cytoplasm, more potassium ions will be there. So these spherical shaped ones are potassium ions shown in yellow color, and hexagonal shaped ones are sodium ions. These are shown in orange color. So outside more orange ions are there. Inside more these yellow ions are there. So inside more sodium will be there. Outside more potassium will be there. So not only these two ions, the remaining ions also will be different, both in the ectoplasm and in the extracellular fluid. So sodium ion concentration will be 150 millimoles in the extracellular fluid, whereas in the ectoplasm, the concentration of this thing is only 15 millimoles. Whereas potassium ions, they are present in the outside only 5 millimoles. In the exterior, in the interior, in the exoplasm, they are 150 millimoles. So remaining things we need not to bother about. These two ion concentrations are very, very important in generating action potential and in maintaining resting potential. So how the resting potential will be generated? So we have seen that this is ICF, intracellular fluid. This is extracellular fluid. So in the intracellular fluid, the potassium ions concentration will be around minus 90 millivolts. It is around minus 90 millivolts. So in the outside, sodium ions concentration will be around plus 60 positive charge. So plus 60 positive charge will be there. So due to the action of these leaky channels, so as I have already told, leaky channels will be there. Sodium leaky channels, potassium leaky channels will be there. So through these leaky channels, what will be happening? Some of the potassium ions will move out of the cell. Some of the potassium channels move out of the cell, and mostly sodium ions will not go inside the cell. So mostly the sodium leaky channels will be closed. So more potassium leaky channels are in open condition. More sodium leaky channels are in closed condition. So more potassium ions go out of the cell than the sodium ions come inside the cell. So only some amount of sodium ions come inside, but more amount of potassium ions go out of the cell. So that's why actually potassium concentration should be around minus 90 millivolts, but as some of the potassiums move out of the cell, minus 70 millivolts of potential will be initiated or will be installed. Or this minus 70 millivolts of condition will be arisen due to the efflux or outgoing of potassium ions. So this is called as what resting membrane potential. So when the membrane is in resting potential, some of the potassium ions move out of the cell more and when compared to the amount of sodium potassium ions coming to the extracellular fluid, only lesser amount of sodium ions get inside the cell. So due to this movement of more potassium out of the cell, less amount of sodium inside the cell, actually minus 90 millivolts of potential should be there inside the cell, but due to potassium ions, some of the potassium ions go out of the cell, it comes to minus 70 millivolts of potential. So this is called as resting membrane potential. So the resting membrane potential is initiated or it is generated due to the potassium leaky channels. Apart from potassium leaky channels, another type of channels also there. They are sodium potassium pump, or it is also called as sodium potassium active pump. Sodium potassium active pump. So the sodium potassium active pump, it is a kind of antiport pump. Antiport means it takes one thing inside and at the same time it sends one thing outside. So when a carrier protein, it sends one ion out of the cell, and at the same time if it brings one thing inside the cell, it is called as antiport. So here this sodium potassium active pump, it utilizes the energy of ATP. Then what will happen? From inside the cell, it will send 3 sodium ions out of the cell, and from exterior, it will bring 2 potassium inside the cell. So 2 potassium ions come inside the cell and 3 sodium ions go out of the cell. Okay, so this will be little confusing to you, and it will be very clear at the end of by the end of this video. Okay, so here what we have said already, that outside more sodium are there, inside more potassium are there. But what this sodium potassium pump is doing, it is sending more potassiums from outside to inside, and it is sending more sodiums from inside to outside. So this will be happening after a action potential is taken place. So once the action potential is taken place, what will be happening? What is the condition inside and outside? When the cell is in resting position first, outside more sodiums will be there, inside more potassiums will be there. Okay, this is clear. When the cell is in resting position, outside more sodiums will be there, inside more potassium will be there. But when the action potential is taking place, then what will happen? Sodiums come inside the cell. Sodiums come inside the cell. More sodiums come inside the cell. That's why only inside potential, it rises from minus 70 millivolts to plus 30 millivolts. So after the action potential is finished, what is the condition? Outside, more potassiums will be there. Inside, more sodiums will be there. Immediately after the resting potential is action potential is finished, inside more sodiums will be there, outside more sodiums, more potassiums will be there. So this is reverse to the action potential condition. Okay, but minus 70 millivolts condition is there inside the cell after action potential is finished. Inside is minus 70 millivolts potential is there, but sodiums are more than the potassiums. Outside, more potassiums are there than the inside. So this thing has to be brought to the again original position. That means again outside more sodium should be there, inside more potassium should be there. So to bring this condition back, this sodium potassium pump comes into picture. It will send more sodiums. 3 sodiums will be sent out. Only 2 potassiums will be sent inside. So after some time, what will happen? Again, after some time, outside more sodiums will be there. Inside more potassiums will be there. This thing will be done all the time, maintaining minus 70 millivolt condition inside the cell. Okay. When the sodium potassium pump is doing this duty of sending 3 sodiums out, bringing 3 potassiums in, when it is doing the total work during the total time of this function, all the time of this function, inside minus 70 millivolts condition will be maintained. So resting potential should be there, but what we need, outside more potassium, outside more sodium should be there, inside more potassium should be there. So to maintain this condition, to maintain minus 70 millivolts condition inside, and to increase the potassium sodiums outside, to increase the potassiums inside, this sodium potassium pump will be helping in this function. Okay, so when the action potential, coming to action potential, so how this action potential is generated? So in oscilloscope, we can see that when the neuron is not doing anything, minus 70 millivolts condition can be seen. So in the oscilloscope, on the x-axis, time is given, time is taken. On the y-axis, potential differences will be taken from minus 90 to plus around plus 40. So till plus 40 or plus 50. So here in this thing, it is shown only minus up to minus 40, but plus 40 plus 50 till plus 40 plus 50 will be taken on the y-axis. So on the y-axis, potential differences, membrane potential differences. On the x-axis, time is taken. So when the neuron is not doing anything, we can see the graph only at this minus 70 millivolt condition. So when the action potential will be generated, when the neuron receives a stimulus. The stimulus could be a touch, the stimulus could be a pressure, the stimulus could be a light falling on the retina, the stimulus could be a sound wave touching this eardrum, the stimulus could be a chemical falling on the tongue, the stimulus could be a chemical falling on the nose. So all these are stimuli. So when the stimulus is given to that particular neuron, the stimulus will, okay, the stimulus will open these things, what we have seen, voltage gated channels or mechanical gated channels or ligand gated channels. So the stimulus could be the neurotransmitter, the stimulus could be the sensation, the stimulus could be the ion of calcium ion entry. So all these are stimuli. So when the stimulus is given, what will happen? This sodium voltage gated channels will be open. When the voltage gated channels are open, more sodiums enter inside the cell. So when the more sodiums enter inside the cell, minus 72, it will increase up to minus 60, minus 50, minus 40, minus 30. Like that, it will go up to plus 30. So when the plus 30 condition is there, it is called as action potential. So potential difference, it is going from minus 70 to plus 30. So when it reaches plus 30, we call it as action potential. That means neuron is doing work. That means neuron is activated by the stimulus and it is sending the stimulus information to the brain, or it is sending the information from brain to what this effector organs. So in this way, action potential will be generated to the stimulus. When the stimulus is given, then only action potential is generated. Okay. So, so here in this thing, all the stimuli are not enough to generate action potential. When we call it is action potential, when plus 30 millivolt condition is there inside the neuron. In the cytoplasm of the neuron, the potential reaches plus 30 millivolt condition, then only we call it as action potential. But all the stimuli will not be enough strength, will not be having enough strength to generate that plus 30 millivolt action potential inside the neuron. So like this way, some stimulus will be very weak in strength, and the second stimulus is little more in strength. The fourth stimulus, it is having some more strength. Okay. So all these stimuli are not having enough strength to generate action potential. All such weak strength stimuli will give or will generate a very less these graphs are these potentials. We call this as graded potentials. Okay, so here in understanding action potential, so far we have understood resting potential. Okay, so in understanding action potential, two other terms also will come in picture. One is graded potential, another one is threshold potential, and final one is action potential. So graded potentials are the ones which are having very weak strength, which are not having a capacity of generating or increasing plus 15 millivolts difference. So when the resting condition is there, minus 70 millivolts condition is there. So minus 72 plus 15 gives minus 65. Okay, minus 65. So, sorry, minus 55, minus 55. So, minus 72, minus 55 will be having plus 15 increase. Plus 15 millivolts of increase is there in between minus 72 to minus 55 millivolts potential. So, this minus 60, minus 72, minus 55 potential is considered as threshold potential. When the stimulus is having enough strength in increasing the potential from minus 70 to minus 55, then only action potential will be generated. So to generate action potential, the stimulus must have a strength of generating minus 55 millivolt potential. So to increase the potential, okay, the threshold potential should have a strength of increasing potential from minus 70 to minus 55. So threshold potential value is plus 15 millivolts. So resting potential is minus 70. When 15 millivolts is given to it, it will reach minus 55 millivolts. When the stimulus is enough to increase the potential difference inside the cell till this minus 55 millivolts, then only action potential will be generated. So this minus 55 is the threshold limit. So when the threshold limit is crossed, then only action potential will be generated. So below this threshold potential, okay, below this threshold potential, all the stimuli will generate very weak signals or very weak potential. These are called as graded potentials. So graded potentials are due to the very weak strengths which are not capable of increasing plus 15 millivolts difference. Threshold potential is the one which is having the capacity to increase the potential difference from minus 70 to minus 70 to minus 55. So threshold potential value is plus 15 millivolts. So below the plus 15 millivolts, they give graded potential. Plus 15 millivolts capacity stimulus will generate threshold potential, and more than plus 15, it will generate action potential. Okay, so these graded potentials. Okay, this graded potential could be positively thing, or they could be hyperpolarizing graded potential. They could be hyperpolarizing, or they could be depolarizing graded potentials also. Two types of things can be seen. So when the potentials are decreasing the polarity or decreasing the potential inside the neuron, they are even decreasing they are further decreasing the potential inside the neuron. Say for example, minus 72, they are decreasing up to minus 90. So the minus condition or negative condition is further increasing due to some signals, due to some stimuli. So such type of things are called as hyperpolarizing graded potentials. So if the potential difference is in increasing position, it is called as depolarizing